action!
Oh hey, didn't see you there. Hi, I'm Professor Millie Molecule from the Central Cell Study Lab in Central South California. And we're here today to talk about cells, and particularly one thing about them. Mr. Ion, my chair please. This is my assistant. His name is Ian Ion, and let's just say he loves to move things, whether it be through a membrane or not. Thank you, sir. Now, let's talk about cells, shall we? Cells are the building blocks of life. We are all made up of millions and millions of these cells, and without them, we basically couldn't function. And so, today we're here to talk about one part of the cell membrane. It is defined here in my biology notebook as the separator of the interior of cells from the outside environment. To elaborate more on that, basically, it's a boundary that allows good things, or like molecules or ions, to pass in and pass out. Or it allows bad things like bacteria and germs to stay out. That way, the cell will not be harmed. So, basically, if you think about it, we can draw an analogy here. The plasma membrane, if the cell was like a building, the plasma membrane would be the security guards because they patrol the outside and choose who to let in and who to keep out. And that's why the cell membrane is so important. In fact, they built a whole model on it, and it's called the Fluid Mosaic model. And we have a beautiful piece of art curated by the one and only me. Come in a little closer so we can see it. As you can see, our artist has created this beautiful 2D model from everyday materials. The phospholipid bilayer was created using ping pong balls and rolled up streamers, while the proteins come from paper tubes, and textured balls were the cholesterol, and the carbohydrate chains were just pipe cleaners. And I think that's wonderful. Let's talk more in depth of about each component of this diverse model. Let's start with the phospholipid bilayer, the base. The phospholipid bilayer serves the important purpose of being a barrier and base for the authorized passage of molecules and ions. So we have a more in-depth model right here showing the hydrophilic heads and the hydrophobic tails. So the heads of the phospholipid are made out of phosphate and glycerol, and they're hydrophilic, or water-loving. And they kind of have to in order to serve their purpose because they're surrounded on both sides by water or liquid. Next, we have the hydrophobic tails. They are created from one strand of saturated fatty acid and one strand of unsaturated fatty acid. And they are lipids. And you know that lipids and water do not mix. As you can tell, if you try to mix oil and water, it doesn't work. That is why, obviously, they are on the inside, so they do not have to interact with water. They're hydrophobic. Next, let's talk about the proteins. The proteins come in many different kinds and shapes and sizes because this is a diverse model, but let's start with the three simple ones: channel, integral, and peripheral. So they help transportation through facilitated diffusion, osmosis, sodium-potassium pumps, etc. Let's start with channel. So we have our channel protein all the way on the far right, and it mainly helps with active transport and facilitated diffusion. Next, we have our integral proteins here and here. They are usually helping facilitate diffusion, and they're also receptors. And finally, we have our peripheral proteins. As you can see, they are on the bottom of the layer, underneath the layer, because they help with signaling and maintaining shape and structure. So, as you can tell, proteins are very important for cell transfer and stuff like that. So, moving the molecules in and out. Next, we have cholesterol, which we can talk about really fast because it mainly just serves a simple purpose: to regulate fluidity in the membrane. So cholesterol hydrophobic tails inside the lipid bilayer, and that is because cholesterol is a lipid, so of course it is hydrophobic. And yeah, that's pretty much it for cholesterol. Next, let's talk about carbohydrate chains. Oh, my voice is getting a little raspy. Ion! Ion! Water, please. Thank you, sir. Anyways, next we have carbohydrate chains. These are very important because they serve multiple purposes such as cell-cell contact, self-recognition, and communication in general. We have the structure of this carbohydrate right up here, and they come in two kinds, glycoproteins and glycolipids. They're glycoproteins if they're attached to a protein, as you can tell. Or they are glycolipids if they are attached to a lipid or a hydrophobic lipid tail inside the phospholipid bilayer. And that is pretty much it for this beautiful fluid mosaic model. I also did a video on my YouTube channel, The Biology Book, and it talks about this same model and its history and where it comes from. So if you want to check that out, go ahead. And this has been Professor Millie Molecule from the Cell Study Lab of Central Southern California. Thank you for watching.